Researchers find links between human and canine brain tumors. This is by Courtney Price from Texas A&M University. Texas A&M School of Veterinary Medicine and Biomedical Sciences, VMBS, Baylor College of Medicine and Texas Children's Hospital researchers have discovered that meningiomas, the most common type of brain tumor in humans and dogs, are extremely similar genetically. These newly discovered similarities will allow doctors to use a classification system that identifies aggressive tumors in both humans and dogs, while also opening the door for new and exciting collaborations between humans and animal medicine. Until now, the lack of reliability and viable experimental models has been a barrier to understand the biology of and development affecting treatments for these brain tumors. The discovery that naturally occurring canine tumors closely resemble their human counterparts opens numerous avenues for exploring the biology of these challenging tumors, said Dr. Akash Patel, an associate professor of neurosurgery at Baylor College of Medicine and principal investigator at the Jan and Dan Duncan Neurological Research Institute at Texas Children's Hospital. It also provides opportunities for developing and studying novel treatments applicable to both humans and dogs. The study, published in Acta Neuropathologica, is led by Patel, Dr. Jonathan Levis, and VMBS professor, head of Department of Small Animal Clinical Services. For the project, the team analyzed 62 canine meningiomas from 27 dog breeds and discovered that the tumors shared remarkable similarities to the same kind of tumors when they occurred in humans. This is the largest study to date of the gene expression profiles of canine meningiomas. And watching the signs, the new discovery was made possible by building on recent work conducted by Patel's team, as well as previous work by Levine and Bordeaux that explored gliomas under another type of brain tumor. In 2019, Patel and others at Baylor College of Medicine and Texas Children's Hospital found that they could classify meningiomas in humans into 3 biological distinct subtypes, MEN-G, A, B, C, by analyzing their RNA. The new classification system can predict patient outcomes with greater accuracy than the standard tissue sample analysis. Because RNA shows how a tumor's gene activates, it allows researchers to actually predict how a tumor will behave, whether it will be aggressive or if it's going to respond to certain therapies, Levine said. In 2020, Levine, Bordeaux, and colleagues at the VMBS and Jackson Laboratory for Genetic Medicine found genetic similarities between gliomas, the second most common type of brain tumors, in humans and dogs. Armed with a new way of detecting aggressive tumors and the knowledge that dogs and humans share some brain tumor traits, Patel reached out to Levine about applying the findings to study meningiomas. We ended up, he said, by agreeing to provide Patel with canine tumor samples we had worked years and years to achieve, to see if he could isolate the RNA, which is not always easy to do, Levine said. He was able to produce this very robust data set that showed a similar pattern structure to human tumors. Our team also provided Dr. Patel with key clinical outcome data, including responses to certain treatments. Moving to clinical trials, now that the researchers established the connection between tumors across the 2 species, they can begin to preparations for clinical trials that can take several years to plan and fund. We're really interested in creating wins for both human and animal medicine, Levine said. For example, we hope to give dog owners access to therapy that's not available anywhere else in the world through clinical trials. At the same time, that information will also inform the next step of human trials. Incidentally, a separate group of researchers from the University of California, Davis, conducted a similar study with matching conclusions about meningiomas in dogs and people and published its work in the same journal. The same research groups look forward to collaborating in the future to develop tumor treatments for both species. Levine said, I think there's a terrific opportunity for the team at Baylor, Texas A&M, and Texas Children's and University of California to collaborate to create a clinical trial, he said. If we do one trial, we'll be able to enroll patients a lot more quickly, which would make it easier to get larger data sets, resulting in stronger findings. So we have a lot of interest in doing the collaborative trial, he said. We really see the team out in California as potential partners. For now, the next step is looking through the data from both studies to see if there are clues that will lead to new therapies. One of the benefits of this project is that we already have all this genetic data and we can use and describe what might take a good treatment, what might make a good treatment, Levine said. Part 1 has set up very well to work on Part 2. And this is by Texas A&M University on Medical Express. Please leave your, by Courtney Price. Please leave your comments. Thank you for your support. Kindly support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.